online, or you can download, and you can save it into your computer systems or in mobiles, and you can watch it whenever you want it. At the same time, this presentation and relevant guidelines documents also will be shared through in email, or I will post in the WhatsApp group. You can watch it or you can use it. So coming to our institute, so institute is a fortunate Veda, so which is biologics, biosimilars, so which is you are using for the trainings, especially for the regulatory affairs in the case of pharmaceuticals and biopharmaceuticals. And this institute located in Hyderabad. So whatever the institute address is mentioned, same address it is registered, and even you can see in the Google also. So you can see in the your website also in the same address will be there. And so this is an ISO certified institute. So ISO QA and ISO 9001 and 2015 certified institute. So that is a recognized one. So your certification, your project completion, internship project completion will be come with your ISO certified institute, which can be valid and which can be in the anywhere you can show it. Yeah, there is no any hiding things here. Institute is a fully open thing. And coming to pharmaceuticals and product development, how it will happen. So if you see in this slide, so there are 5 phases. So how it will happen? So a drug product, it may be chemical drug product, it may be biological drug product. How it will development will be start? So development will start with a discovery phase is nothing but R&D. So the research and development, which is R&D people will be initiate research on the new chemical moieties or generic drugs or biologics or maybe biosimilars. So they will be working on this discovery phase, and they develop the at small scales, and they will identify. So this is the suitable drug, and this is the generic drug, or it may be biosimilar drug, which is equal to innovator drug. And after confirmation, so before starting of R&D, we can't go easily and start it R&D activity. So before starting of R&D, so we should get test license. So that is the regulatory affairs, a person job role to get that test license from the regulatory body. So regulatory body in the sense we are from India, so we are going to approach with our CDSO board, and we are going to get it a permission from them. So once we get permission from them, we can start R&D. So there is a, so in that under that board, there are called RCGM. I will explain what is that RCGM. And there is a IBSC board who will give the permission to us to use any biological organisms, any harmful organisms going to use for the experimental purpose. So we should be intimate them, get the permission, then only we can start it R&D. So in R&D, what will happen? Cell bank constructions, and followed by upstream process development, downstream process development, and followed by formulation development, analytical development, and all the developments will be optimization. And at the same time, in chemical drugs, so synthesis. So synthesize the new chemical moiety, entity, new chemicals, or it may be generic drugs, or maybe identical generic drugs. We are going to change in the formulations, will be make it R&D trials, and will confirm the drug is equally or optimized the process. So once it is done optimization, it is a chemical drug or it is a biological drug. So we want to conduct preclinical trials on the animals. So when you go into preclinical trials, so in case of new chemical entity and new biologics required to conduct on animal trials. So where in case of generic medicines not required to conduct. The reason is generic medicine is a identical molecule of the your innovator product, which is branded chemical moiety. In case of biologics, so when you're going to make a biosimilar product, so the biosimilar product after optimization is required to conduct preclinical studies. The reason is this is not the biological molecules are complex molecules, large molecules, and it's not the identical molecule to innovator product. It may be slight changes due to the large molecule capacity, as well as it is a complex molecule and it is coming through living things. So there is a slight variation, so that the variation can be accepted. So required to conduct safety studies on the preclinical studies by taking model animals. So in this model animals, what type of animals will use and how long it is to study? Is it required to single dose, or is it multi-dose, and safety precautions all will be studied here. So we can't start directly animal studies. So what we'll do after completion of optimization of R&D, so we'll take optimization data and we'll show to the agency. Agency in the sense in India, so we'll approach to IBSC board and especially for the biologics, and we'll go for RCGM review. Upon review and approvals of protocol, so we'll get the approval of the protocol and we'll go for conducting preclinical studies on the animals. Once you conducted preclinical trials on the animals, and upon receiving the reports, and all the reports, if complies against your protocol, so there are safety studies was complies whatever the specification is mentioned, it is meeting the criteria. That means your preclinical application is completed. So the reports are completed, and the reports are going to submit to the agency to get the recommendation to go for clinical trials. So once you submit this preclinical reports, agency will be review, and upon review, they will give queries. If anything gaps, if anything not filled against your protocol, anything deviating from the protocol, so where it is required to provide a justification or required to provide a additional data when agency is asking against protocol, against submitted data. So those are all going to do by your regulatory affairs team, regulatory affairs people. So during your internship, if you take one of the case study, how to get the application pathway for R&D activities, or how to apply the preclinical trial application, and how to submit the preclinical reports, you will come to know one of the topic. Either you take R&D application or you can take preclinical application. Next, so once preclinical approvals is completed in India, so the next step is the manufacturers. The manufacturers looking for Indian market or looking for Saudi FDA, or maybe looking for ROW market, FDA, EMA, or MHRA, or maybe Health Canada. They like that different regulatory bodies are there. So which country you are looking for it? Are you looking for country specific like US FDA, regulatory market? Are you looking for region specific? Yes. Yeah. So are you looking for country specific submission, or it is a region specific submission, or it is a global filing? So RLB markets also can possible. So based on that, your investigational new drug application to be placed. If you are looking for domestic market, India, so this investigational new drug application to be submitted to the CDSO board to get the permission to start clinical trials on the humans. Or you are looking for the filing activities in the US FDA or maybe in the EMA, the IND filing should be done in the specific country or specific region to get the clinical trials protocol approval to conduct on the human studies. So of new chemical drugs or new biological drugs, or is maybe biosimilars or maybe generic drugs. So the clinical trials phase 1, phase 2, phase 3 is applicable for regarding your new chemical entities and responsible required for the new biologics drugs. In case of generic drugs, not required to conduct clinical trials because those are the identical molecules, identical products. So there is not required to conduct preclinical trials and clinical trials for the generic drugs. In case of biological drugs, for the biosimilars, required to conduct preclinical trials. In case of clinical trials, so there is phase 1 is required to conduct and phase 3 will go to conduct. In case of biosimilar drugs, and phase 2 is not required. Why it is not required? I will explain coming slide. So, based on the approval of the country and business strategy, our organization our organization is looking for Indian filing, organization looking for MHRA filing, UK filing, or looking for Saudi FDA market, or maybe looking for US FDA or looking for EMA. So based on that, our regulatory affairs team will be filed this product for the filing into the respective country or respective region to get the approval for clinical trial conducting. So once you got protocol approved, so the batches which is optimization done at R&D, the technology will be transferred to GMP area. GMP means good manufacturing practices in the manufacturing area in our organization. So we are going to take clinical batches in the GMP area. So here, how technology transfer from R&D to manufacturing, that is CMC data, and how analytical methods were transferred from R&D to your QC laboratory, that is a CMC data. So that is not all we're going to learn it. So that is also one type of project internship can be take up tech transfer. And once clinical batches is taken, and after that, so clinical trial is going to conduct it. So your phase 1, phase 2, phase 3, wherever applicable, that will be conducted, and protocols will be prepared separately, and execution will be done, and reports will be collected, and the reports will be going to file into new drug application. In case of USFDA, or you are going for EMA, marketing authorization application, will go, going to apply it. In case of USFDA, for biologics will be BLA, biological license application. In case of new drugs, we'll go for new drug application. In case of generics, we'll go for abbreviated new drug pathways. So like this, like that, based on the type of drug you are going to file, based on the type of country, the terminology will be different, but way of filing will be almost all similar. Where you are going for EMA filing, where you are going for FDA filing, where you are going for Indian filing. So the data which has to be generated, so that data can be support for any type of country. Only few things administration point of view, different country to country, region to region, and country to country. But your technical data will be remain same. So after completion of clinical trials, so we'll submit the all CMC data and clinical data, preclinical data to the agency through new drug application or marketing authorization application. So here also the regulatory people will be involving collection of the data, review of the data, preparation of the dossiers, and submitting to the respective filing pathways. So after submission, the reviewing process will be initiated by respective regulatory bodies or regulatory agencies or regulatory consultant also, we can call them. So after review, so they may be issuing queries, or they will come for our plant visit audit, nothing but audit, GMP audit. So GMP means good manufacturing practices, GMP certification to be given to the respective manufacturing area. So after completion of your audit, and after completion of your audit, followed by you are going to get an approval for the particular product, and you are going to file for the WHO certificate, WHO GMP certificate, and we are going to take the batches here. So the process validation batches, and upon the batches of drug substance, followed by drug substance, followed by drug product. So we are going to place for stability, and after showing the stability for 6 months, so we'll get the WHO GMP certificate. So that is also going to handle by a regulatory team, the regulatory people. So that is also you can take up one of the internship project, how to file the product to GMP application for the getting the product into launch in the market. That is also one of the session, one of the topic being selected. And once product is launched in the market, and our manufacturers will be continuously manufactured, commercially distribution is happening. So that means your drug, whether it is a chemical drug, whether it is a biological drug, so continuously being distributed in the market, and patients being are using in the market. So either it may be country specific, or maybe worldwide the drug is commercially distributing. So when commercially is being distributing, so there is in clinical phase 1, phase 2, phase 3 already shown the data, and commercial, when there is a commercial distribution happening for the period of 2 to 3 years, we have to collect phase 4 data. So there is a phase 4 data protocol also to be shown before launching the product. So this is also handling by your regulatory team people. So after commercial distribution, we'll collect the phase 4 data. And meanwhile, if you are going to change in the process, so the manufacturing process, manufacturing site changes, or any equipment changes, site changes, anything happening, we are going to handle through post-approval changes. So the post-approval changes also one of the internship project you can take up. So like this, we have many topics to in different stages. So it's not possible to start to end one internship program can take up. So our course can be conducted how to initiate R&D, how to reach to the market, how to take post-approval changes will be continue. For you people, you can take up, there are end of the session, I'll show what are the topics for suitable to internship. You can choose any one topic and you can start studying, and we'll guide you, and we'll take the sessions around 45 hours. In that 45 hours, you will get knowledge from R&D to till market stage, how to reach the market product, product is reaching to market. That is what going to teach, that is what going to learn from this program. Yeah, so continuation of this slide. So is it clear to everybody? Any questions do you have here? I can spare 1 minute time. Anyone can talk. Yeah, please unmute and you can ask any questions. So this is the time where you are spending 1 hour time. this should be useful for you. So there should not be wasting of time of 1 hour, whatever you're spending here. Yeah, you can unmute, you can talk. Yes, Afifa, do you have any questions? Megharaj, do you have any questions? Is my voice is audible to everyone? Yeah, please.
Hello.
Please.
Sir, this course is a paid course.
This session is a paid demo session, and the 45 is the paid course.
Okay.
Yeah. Yeah.
So you will provide a placement assistance also means.
Okay. Placements we will not be 100% assurance, but 100% assistance will be there. So we'll guide you. I will tell you end of the session you will come to know. Just that will be end of the session we'll discuss. Technical point of view in this doubt is there, let me know.
Okay.
Okay. Any doubts to anyone in this slide? Hope everyone is following with me, right?
Sir, so different filing is there for UK, US, Canada, and other
Welcome.
countries.
Yes. Exactly, available.
Okay.
Yes, Darshan, do you have any questions? Okay, fine. Yeah. So, yeah, please. Okay, fine. See, so the dossier will be a common technical document in generally we'll call as a CTD. So how the CTD will be aligned? The CTD will be like module 1, module 2, module 3, module 4, module 5. So what are these modules? So these modules are when you want to file as per CTD in the ACH regions. So the dossier should be prepared in these modules. So module 1 is a non-technical documents. That means regional administration information like licenses we have to submit, and related to patents, related to any administration point of view, country to country, the requirements will be different. So this is what this model was mentioned as a not part of CTD. And part of CTD is model 2, model 3, model 4, model 5. So when you model 2 is a index kind of thing for the quality module, so which is model 3, and model 4, model 5. So model 3 is a CMC, chemistry manufacturing. So this module will cover from your R&D, your clinical batches, your commercial batches, and your process validation, everything will be covered. And here the entire quality data will be there. It will tell that what is the purity, what is the impurity levels, what is the potency of the drug. And coming to module 4, which is non-clinical study reports, nothing but animal studies. So which we are conducting on the animals for the safety studies. So this will be reports will be enclosed here and respect to module 2, non-clinical overview, non-clinical summary to be provided in the module 2. And coming to module 5, clinical study reports. The clinical study reports related to clinical overview, clinical study will be provided here. And this is what a module 5 in the CTD triangle. So in this CTD triangle, we'll cover module 1, module 2, module 3, module 4, module 5. So in our course, we'll be cover all the modules, how to prepare it, how to file in the respective MOH. MOH means Ministry of Health. So this module will be having all technical information. And continuation of this, molecules like if you see there is a aspirin, a simple pharmaceutical chemical moiety, which is chemical drug, a small molecule. And when you're talking about chemical drugs like insulin, erythropoietin, or monoclonal antibodies, these and all will fall under biological molecules. So these biological molecules are too complex compared to chemical drugs. If you see here, the molecular weight is mentioned here in the Daltons. Aspirin is 180, and insulin was 5,808 Daltons. The size is too much difference. And erythropoietin, almost all 5 to 6 times more than insulin. And monoclonal antibody, almost all 5 times more than erythropoietin. So where is 180 Dalton of aspirin, and where is monoclonal antibody 150,000 Daltons. So this is what the aspirin like molecules can be identical molecules. We can produce same molecule if you manufacturing in India or if you want to manufacture in the US. But insulin, erythropoietin, monoclonal antibodies, these are the biological drugs which are very complex. And if you manufacture in the same facility also, there is a slight variation will come. So that is the reason these are the comparable molecules, not any identical molecules. So when you're going to make a biosimilar drug, so innovator product versus your product will be slight variation will be there in the point of purity, in the point of impurity levels, in the point of potency, in the point of identity. There is identity will be remain same. So there is some certain parameters, certain attributes. There is a certain changes will be there, but those changes may not impact the potency of the drug, efficacy of the drug. That has to be proven in the clinical trials and preclinical trials, and as well as biosimilarity, complete biosimilarity assessment. So complete biosimilarity assessment in the sense, say a molecule from analytical comparability, preclinical comparability, and clinical comparability to be proven the innovator product and this biological product are same in the point of view purity, impurity, potency, that has to be proven. So a pharmaceutical product versus biological product. So the size of the molecule which is less than 1000 Dalton, which will be fall under chemical drugs. The biological drugs greater than 10,000 Daltons. So those will be falling under biological sections. The source is chemical synthesis. These are coming from chemical. So this is from living cells. So by using chemical synthesis products, there are side effects. There are many side effects to the people. Maybe not today, but maybe later in the stage of life cycle management, there are giving some side effects. But when you're going to use living cells for producing of your for the medicinal products, it may not be having that much impact on the side effects. So that is what the chemical drugs are replacing with your biological drugs. Slowly, the biological drugs will be rule the entire world. And form, generally oral or solids will, or maybe injectables will come in the form of route of administration. In case of biological drugs, so it will be coming through injections or infusion through intravenous systems. So this is coming subcutaneous or maybe intravenous systems. It will be route of administration will be happen and dispensed by usually retail pharmacies. There is no any prescription also required or may not require, may require or may not required for dispensing of these medicines by pharmacists in the stores. But in case of biological drugs, definitely required recommendation from doctor prescription. Then only this will be dispensed. So like there is an example, Lipotor, one of the chemical drug, a small molecule, 558 molecular weight. In case of there is a Herceptin, which is cancer therapy drug which are using for breast cancer treatment, and almost all having 185 Dalton of 185 kilodalton of molecular weight. So this is biological drug, this is a chemical drug. So this is the huge difference of this. And coming to continuation of this, so like for example, insulin, how mechanism will be naturally happen. If you see in this picture, so when a normal person, how the insulin control in our body, you may see there are patients suffering with insulin diabetic conditions. There is a imbalance in the glucose level in the body. How that is imbalance will happen. So if you take a mixed meal of carbohydrates plus proteins in our body, so when it will go for digestion and will be intestine will be digestion and followed by the product will be in the glucose form in the release in our body. The glucose molecules will be distributed throughout the body. So the glucose molecules when reaching to respective cells receptors. So the cells has to be take up. The cells has to be take up. This is the cell which is the glucose receptors. There are glucose receptor. To receive this glucose molecule, the glucose receptors, there is a insulin has to be code this. The insulin molecule when come and code this, then only the port will be open for that particular cell. Then only the glucose molecule can enter here. So where is this insulin is producing? The insulin is producing in our body, one of the organ of the body, pancreas. In that pancreas, there are specialized cells, beta isolates. That beta isolate cells will synthesize insulin. So due to genetical disorder or due to aging of the body, so the insulin synthesis by beta isolate cells in the pancreas sometimes will get fail. So or maybe dysfunction. So when there is a fail to synthesize or partially fail or completely fail beta isolate cells to synthesize insulin molecule, so what will happen? The insulin will be shortage or maybe insulin will be absent in the body. When there is a insulin absent in the body, these cells, when glucose is reaching to this place, the cells is no receptor signaling of insulin and there is no stimulating insulin, so that there is no take up of glucose. When there is no take up of glucose in the body, so automatically our blood sugar level will be increased. Blood sugar level will be shoot up because of lack of insulin. There was no consumption of glucose. When glucose not taking up by body, our body tissue cells, so automatically there is a no energy formation. Glucose enter into the cell and their complete glucose mechanism will happen. It will generate ATP, adenosine triphosphate energy. So when there is no energy formation, our body will be suffering with lack of energy, nervousness, and when there is a shortage of ATPs, many other organisms also stop functioning and it will be fall under sick. So automatically the person who is surviving regularly, normally, he will become as a sick and he will become as a patient. At the same time, when glucose level increases in the blood sugar level is shoot up, so it will cause other diseases also. So slowly other organisms also fail to function. So how we are going to control it? So when there is a natural insulin is failing in our body or partially not functioning in the body, how we are going to control it? So earlier days, there are chemical drugs to use it to control the glucose mechanism. Now, biological drugs is coming into place like insulin glargine, insulin lispro, insulin aspart, so insulin human. So those are the drugs delivering through biological systems. We can grow the cells and we can produce the respective biological drug, and the drug can be given to the patient body. And when you inject into the subcutaneous injection, so the insulin molecule will be reach into our body and it will be distributed through fluid systems, through blood circulation or to the body fluid systems. And wherever there is a receptors is there, the which is injected through injection, that insulin will be stimulate to take up glucose. So that is the reason the patients who has been identified as a diabetic patient. So prior to taking this meal, the patient will be injected with insulin injection. So it may be insulin human, insulin glargine. There are many type of doses. So based on the type, so we can inject the body according to is it required to before meal or once in a day. So based on that type of injection we're going to take up. So before taking a meal, inject the body with your insulin and take the meal. So what are the taking a meal? It will go for digestion. It will release the glucose. The released glucose mass will be taken up our body and body will be consume it through insulin which is present on body. So that there is no any challenge, lack of insulin and failure of synthesis ATP and glucose molecules are shooting up in the body. There will be not going to happen. If injection is not given, meal is taken, there is a challenge for increase of your blood sugar level. So immediately patient has to go taking off injection, then only he can able to survive. Otherwise, he will become a further diseases or maybe not able to survive in the as usual normal. So that is the reason injection is required. Now, how to produce that injection? That is what we are going to learn from here. So this is the what are the explain. That is the mode of mechanism, how insulin is going to react in our body and how the glucose molecules will be converted into glucose 6 phosphate production so that it will be survive for the body energy. Yeah, continuation of this. So the drug may be biologics or maybe biosimilars. So what is the biologic? What is the biosimilar? We should get the understanding here. So a biologic drug is nothing but so which is coming through living things. That means it may be E. coli type of organism or maybe mammalian cell culture or maybe yeast or maybe fungal or what are the type of organism we are going to use for production of your biological medicine through living cell. So that is called biologics. It may be vaccine, it may be monoclonal antibodies, it may be insulins, or it may be any type of biotherapeutics. Even blood products also will be called biological products, or it is coming through tissues, or maybe through blood plasma products. Those also will be under biologics. So the reference product, what it mean by reference product? So a reference product is nothing but which is registered in the first time in the FDA, Food and Drug Administration in the US FDA, which is registered under first time. So there is a pathway to how to register biological product in the which is innovator product or a reference product. So there is a called biosimilar product. What is meant by biosimilar product? A copy of the biologic against this reference product. So exactly equal to against this product, we are going to make a biosimilar product. This is called as a biosimilar product. So when you are going to call it as a biosimilar product, so when you file in the US FDA market and FDA market has been reviewed your data of CMC data, of your clinical data, preclinical data, and approved this product in their market, then it will be called as a biosimilar product. Biosimilar product. So when it is approved in the market, so the doctor can be prefer either reference product or can prefer biosimilar product. When he is written reference product to be given to the patient, he has to use reference product. When doctor is advised to use biosimilar product, we can use biosimilar product. The cost wise, just imagine this may be $100, this may be $20. Just imagine in your Indian currency, this may be 1,000 rupees, this may be 200 rupees. Only 20% of this product. So that is what the agencies, nothing but government authorities, always look for biosimilar product or generic drugs in case of chemical moieties, so that this will be cheaply available and patients can be easily buy and easily can be use it. So this is already approved by agency. That means it is equally to this product. So it is analytically similar, biologically, but total biosimilarity is similar to this reference product. It is already proven. Now coming to interchangeable biosimilar. So this is also one of the biosimilar like this product, but having additional studies in the clinical. So which is called as a interchangeable product. So interchangeable product is a biosimilar product that meets additional requirements. So additional requirements, especially in the clinical trials, will conduct substitutional studies with your reference product. So this interchangeable biosimilar can act as a equal to your reference product, or when doctor is preferred reference product, the pharmacist can be replace your in case of it is not available in the market. So even though the pharmacist can be dispense instead of reference product, he can dispense interchangeable biosimilar. So the patient last 3 years, last 4 years being used reference product, and it can be replaced with your interchangeable biosimilar. So that much confidence can be given by through interchangeable biosimilars. So interchangeable biosimilars is like there is a insulin glargine which is registered in the FDA market, which is the manufacturers Biocon-Biologics is making as a in the brand name of Semically. So that is the product which is got as a interchangeable biosimilar product. So registered under FDA market. So today they are supplying to FDA approvals. Yeah. Is it clear to everyone? Any doubt here? Hope everyone is following with me.
What is interchangeable product once? Can you repeat?
Yeah, interchangeable biosimilar is nothing but this is also one of the biosimilar, but this is having a additional studies in the clinical. So there is a called additional studies. There is a substitution studies. When you conduct substitution studies, so this product will be approved as an interchangeable biosimilar. When it is approved as an interchangeable biosimilar in market, in medical store, even it is not available, even the doctor is preferred to use this one. So there is not available reference product in the stock. So there is available in the interchangeable biosimilar. Without approaching to doctor, we can give instead of reference product, we can give interchangeable biosimilar product to the patient. So this is having additional substitution studies data, which is don't have by biosimilar product. So that this product will be value will be more than biosimilar product. So that is called interchangeable biosimilar.
Okay, I got
it.
Thank you.
Yeah.
Thank you.
Yeah. So now design and development of biologics and biosimilars. So a biosimilar, a biological medicine product, which is an identical copy of original product of different manufacturer company, is called as a biosimilar product. So biological drug contains substances that have been created by using living cells or organisms. Examples: gene therapies, transplant tissues, or recombinant proteins, or stem cell therapies, and MABs. Those are called biologics. So if you see here, biologic drug and your biosimilar drug. Yeah, please. Akash.
Basically, they are vaccines or means vaccines we can call them biologics.
Vaccines will be
serums
under biologics only, not in the term of biosimilars.
Okay.
Yeah.
Okay.
Yeah, fine. So, so that will also cover in our session, our regular sessions. Vaccines also one of the case study. Yeah, fine. So biologics are biosimilars. So both are identical. So the reason is it is registered first, and after completion of the patent expiry, the manufacturers are going to register with our biosimilar drug. When biosimilar drug is approved, the patient can be, the doctor can be prefer either biologic drug can be used or biosimilar product can be used. The function of the both are same. So if you're going to use this medicine or this medicine, the function will be remain same. Route of administration will be same, and function of the mode of the function also will be remain same. So the cost also difference biologic drug versus your biosimilar drug, so that will be easily can be by the patient and easily can be available in the others, so that we can save many human lives. So that is what biosimilars are playing a major role, and agency also look for the biosimilar products. Yeah. So these are the products like whatever I discussed. There is a teriparatide, so one of the molecule, insulin molecule, and there is a MAB, monoclonal antibodies. So if you take MABs, today there are many patients are saving life from dangerous diseases like cancer therapies. The cancers which is chemotherapies by using monoclonal antibodies. So this will be completely, we can come to know in our regular studies. Human insulin is the one of the case study for the CMC, and our monoclonal antibodies one of the CMC study, and our insulin vaccines also one of the CMC study. So these are the molecules we know how to create the structure and how to preparation of the cell banks, how to do the upstream culture processing, how to do downstream purification to get the pure monoclonal antibody from the impurities, and how to characterize this as a part of analytical characterization, and how to formulate this, what are the excipients you are going to add it, and how to make the innovator product equal to your composition. These are all going to learn it like this molecule preparation in part of our 45 hours regular course. And continuous of this, DNA cloning, how we'll do. So this is also cell bank preparation, part of cell bank preparation. So as we discussed, the insulin is the case study. When there is a body is failed to synthesize insulin molecule, so we have to identify what is the gene of interest is responsible for synthesizing the insulin product. So cloning, vector identification, and your the eukaryotic chromosomes identify the gene of interest, separate that gene of interest, go and create a vector system. So in that gene of interest will be create a vector system. That vector system will be placed into a strain, which is called, it may be E. coli, or it may be eukaryotic cell, or maybe prokaryotic cell, will be placed based on the platform we are going to select it for manufacturing. So we'll place that vector. When it is placed in the vector in the body of the cell, so that will be when cell division takes place, this vector also will be divided, so that multiplication will happen. When there is a biological pathways like protein synthesis is happen, so automatically the gene of interest will code for to synthesize the respect to insulin, respect to monoclonal antibody, respect to recombinant vaccines. We can able to produce it. So after production of these cells will be take it, that cells will go for harvesting. Like this is a one of the cell which is identified cell and placed into vial, and that vial is inoculated into flask, and the flask is culture is grown and transferred into seed flask and followed by production bioreactor. So we'll produce large number of, it may be 1,000 liters, 5,000 liters, 50,000 liters, 1 lakh liters. Will be produced into large number, and we'll go for downstream purification. So downstream purification will use chromatography techniques, filtration techniques, and will purify the respective MAB, monoclonal antibody, respect to vaccine, respect to insulin molecules, and we'll go for formulation. So the formulation will make it into, it may be cartridges, it may be vials, it may be pens, or what are the equal to innovator product, or maybe new biologic, you can create your own container closed system. So which will be it will be mode of action or mode of route of administration easy to give to the patient. So that final drug product will be filled into vial or cartridge or pen and will be commercially distributed. This is what final product will be make it. In our 45 hours course, we are going to discuss complete maps, complete insulin molecules, complete vaccine manufacturing. I will do start with cell banks, end with your drug product preparation. That will be covered, so that it will be helpful for you, part of your regulatory job searching, or maybe part of your process, or maybe part of R&D jobs, or part of manufacturing jobs. So it will be give lot of knowledge to get it idea, so that after completion of this internship project, you can easily can be answer any type of interviews when you want to enter into biopharmaceutical industry or in the pharmaceutical industry. Yeah. So generic drugs. So far we discussed about regarding biological drugs. Now we are going to discuss about generic drugs. So generic drugs, like there is a patents. There is a of the patent we are going to make it. There is a branded name drugs and there is a generic drugs. Branded name drugs, this is the first one registered in the market. So this is a under patent protected. When coming to generic drugs, there is no any patent. Then trade name. Coming to branded names, marketed under unique and proprietary name given by the company protected. In case of generic drugs, marketed under generic name of the drug. So coming to manufactured by, so developed and manufactured by an innovator company. In case of generic drugs, manufactured by several pharmaceutical companies with their different names. And animal and clinical studies, as I mentioned initially, it's not required. So it is a not required. So there is a in case of branded drugs, it is a required to conduct all 1 minute. So all phase 1, phase 2, phase 3, and as well as preclinical studies required to conduct. Animal studies, clinical studies. In case of generics, not required. Price. These are the costly than generic drugs, and generic drugs very cheap, as I mentioned. Similarly, biosimilar drugs is cheaper and your innovative products are very costly. And appearance. So unique look designed during product development. And coming to generic drugs, look different from relevant brand name drugs. And the name variations. In case of branded name drugs, same or different brand names in different countries. In case of generic drugs, same generic drug name in any country. So this is the differentiation between branded drugs. This is the generic drugs. Why it is cheap means, why it is costly means. So the manufacturing cost for the development, for the manufacturing, for the conducting clinical trials, preclinical trials, here the manufacturer is required to invest a huge money for development and clinical studies. But in case of generic drugs, there is no animal studies, there is no clinical studies. Hence, the manufacturing cost will be less for the manufacturer. That is the reason it is a cheap. At the same time, here many people will be in competition. Here there is no people in the competition. At the same time, the manufacturer of the new chemical brand name drugs is invested a lot of time. Maybe 10 years, maybe 15 years, he invested a lot of time to get the complete all studies, get the approvals from the agencies. Then he was given a certain time period for 5 years, 7 years time to sell this product in the market. Upon completion of this patent expiry, the generic drugs was trying to file into the respective country of origin or country of distribution or country of filing, so that they will have less cost compared to innovative drug. That is the reason these are the cheap, these are the costly. Yeah, is it clear? Generic versus brand name drugs. Hope everyone is following with me. Yeah, please respond.
Yes, sir, get it.
Okay, fine. Yeah, please respond. Whenever I'm asking, please respond so that I can understand if people are following with me. Others hope following with me. Yeah, you can text also. Yeah. So as I discussed, prescription of drugs cost comparison. Generic drugs, if you imagine 25 rupees, branded name will be like 85 rupees. The reason is, if you see some other drugs also, paracetamol, there is a generic drug 2 to 3 rupees per tablet. In case of branded drugs, 11 to 17 rupees will be there. Similarly, paracetamol syrups, any diclofenac, any amoxicillin, azithromycin, folic acid, B complex. The difference is huge, almost 20% only. If you take this the 10 rupees, this will be around 2 rupees. So the differentiation will be around 15 to 30%, where it will be there price wise. But when you take generic drug for the any diseases, any control of your body, or you take branded drugs, the response when you're going to check in the plasma in our body, the response will be remain same. The peak response is increasing this nanogram per ml in the plasma concentration. Again, it is coming down by time being 24 hours in the same profile. By seeing this, is there any difference? Can you tell someone? By seeing this structure of profile of the chromatogram, plasma concentration when injected in the body through subcutaneous or maybe oral any medicine you taken it, when you go on to test the plasma concentration or blood concentration, the presence of your drug, the certain drug, so which is showing the concentration response almost all similar. Is there any difference? Yeah, please respond.
Yes, sir, difference is there in mean plasma concentration.
So how much differentiation is there? Around 65. This is around 68 something, correct?
The
Yes,
time of
sir.
response is early in generic, sir.
So the difference is not much. If you see, this is 1 hour. If you see, it is reaching in 1 hour. It may be reaching 2 hours. It may be reaching 4 hours. The slight variation. But when it's coming down, the response will be almost will remain same. At one point, it is to same time. So if you take for the particular disease, if you take generic drug or if you take branded drug, so both will be the remain same. There is not much.
In broader sense, it is okay, sir.
Yeah. So overall, if you see, both response are same, correct? So the reason that is the reason, either you take generic drug, either you take branded drug, up to you, or maybe any differentiation there. The physician will be suggest to take either this one or this one. So accordingly, we can go ahead. But the filing pathways, so which we are going for generics will be different pathway, which we are going for branded medicines, which will be different pathway, and the data will be different here, will be different here, but the same data will be remain same, but your clinical and preclinical data will be different, this to this. That is what we are going to learn from continuation of regular course. Yeah, continuation of this, the guidelines. There are many guidelines, so which will be part of your manufacturing in case of biologics. So there is a ICS guidelines. There are 14 guidelines. Under that, there is 14 guidelines. There is a Q1, which will be talk about stability. Q2, method validations. Q3, imperatives. Q4, related to pharmacopoeias. Q5, biotechnology guidelines. Q6, related to specifications. Q7, your GMP guidelines. Q8, pharmaceutical development. Q9, risk assessment, pharmaceutical risk assessment. Q10, pharmaceutical systems. Q11, development. Your Q12, Q13, Q14. Like this, there are guidelines, 14 guidelines, which is referred for your development of your process development as well as your filing pathways. So these all guidelines we are going to refer coming days, which can be help you for part of internship, part of project preparations, part of your process development, part of interview preparations. This can be help you to these all guidelines. When you know the guidelines, easily can be face the interviews. In definitely, when you're going for interviews, they may ask what are the ICS guidelines. So when you attend carefully these sessions and when you prepare any internship project, internship program, you can come to know. You can come to know what are the guidelines, which guidelines are responsible for, and which stage. That can be come to know. Yeah, this will help you in the total guidelines. And not only limited with ICS guidelines, we are going to refer WHO guidelines, going to refer Indian guidelines, going to refer other wherever the required guidelines will be going to refer in respect to country specific, respect to region specific guidelines. Yeah, so roles of different authorities in biologics biosimilars licensing. So if the key regulatory bodies, like there is a Drug Control General of India, which will be mainly authority clinical trials, ensure standard clinical trials purpose or any registers all imported drugs, new drugs or biologic drugs, medical devices. So these are the department is responsible to register and review and approval. So coming to Central Licensing Approving Authority, which is CLA, is provides approval for manufacturing licenses. And Central Drugs Laboratory, CDL, National Control Laboratory, which is responsible for testing of your drugs. So what are the we manufacture, they will test and they will confirm this drug can be released or can be hold, can be reject. They will verify it, the data. Department of Biotechnology, DBT. So overseas development and modern biology and biotechnology in India, especially in India, there is the department is responsible to ensure for the biological products. And review committee for genetic manipulation, which is RCGM, for the recombinant products. So natural strains, DBT itself can be taken care, but when it's coming to genetically modified organisms, so which is genetic system is modified. So RDNA products, especially RCGM is the responsible to review and approval process. Indian Council for Medical Research, especially for the formulation studies. So this is the department is responsible for taking care of all formulations related to drug product related one. And Genetic Engineering Approval Committee, GEAC, is responsible for all commercial activities and our environmental protection. And Drug Technical Advisor Board, so which is providing a technical guidance to the CDSO. And National Pharmaceutical Pricing Authority. This is the authority in India will be decide what cost to be given to the respective medicines, which is registering under government authorities. So to be decided by price cost. Based on the manufacturing cost and based on the competition, price will be decided. State Licensing Authority, each and every state, though CDSO is the main board and central of the central government is located in the Delhi, and every state like Telangana, you take in Hyderabad, there is a state licensing authority. Karnataka, you take it in Bangalore, there is a state licensing authority. Or you take it Maharashtra, take it Madhya Pradesh, every state will have a local authority. So responsible for licensing of manufacturing site, drugs, establishments and approval of your drugs, testing laboratories, monitoring, qualifying, and drug cosmetics and marketing in India, and recalls substandard drugs will be taken care by state licensing authority. Continuation of this acts and rules. So as per Drugs and Cosmetics Act in 1940 and 1945, regulated drugs manufacturing, quality and marketing of biologics and biosimilars in India, there was a specialized guidelines, what to follow, what not to follow. There is a guideline is called Schedule M for good manufacturing practices. And there is a Schedule Y for conducting toxicology studies like animal studies and clinical trials and import and manufacturing of new drugs. So to be followed, Schedule Y guidelines. And new drugs clinical trials as per 2019, there is a Schedule Y guidelines and are now replaced with your new drug clinical trial applications. And Schedule M guidelines also recently get revised. and CDSO guidance for industry for approval of your clinical trials and marketing authorization. So this is the centralized body who is the Indian regulatory body is taking care of all these activities and especially for the approval of clinical trials and marketing authorization. So RCGM and Department of Biotechnology will take care of initial role for the approval until preclinical. And after preclinical completion, clinical and marketing authorization is taken care by CDSO board. And CDSO guidance for post-approval changes. So already approved product want to change something changes, then there is a CDSO guidance for post-approval changes, separate guidelines need to be referred and need to go through variation filing system. And pharmacovigilance for the document Indian Pharmacopoeia Commission IPC guidance for manufacturing and PV practices following during your clinical development as well post licensing. So how you wanted to follow all pharmacology to be followed this guideline. And WHO guidelines as a part of manufacturing required to know, and you part of other product specifics, any TRS technical reports required to follow, and regulations and your guidelines for recombinant DNA research and biocontaminants, Department of Biotechnology guidelines, as well as RCGM guidelines to be followed for the biological drugs. Yeah. So a manufacturing process development PowerPoint table if you take it. So here manufacturing overview, project management, development under R&D, project requirements, product development support, process development. Then we'll go for manufacturing. So custom manufacturing, support services, global distribution. When you're talking about global distribution, including outside of the India, not only limited with India. So it will be throughout the world. We can start global manufacturing followed by global distribution. When you want to global distribution, you should have filing authorities, filing pathways, and you should have a overview on which country you want to go. Accordingly, file that product in that respective country. After approval only, you can start commercial distribution. And the products like RDNA products, biosimilars, biobetters, and novel biologics, vaccines, new chemical entities, generics, medical devices, in vitro diagnostics, and clinical trials, narcotic drugs, psychotic drugs, veterinary drugs, drug pricing, Unani Ayurveda, and cosmetics, herbals, and cell and gene therapy, even food products. These many products are falling under Indian MOH, Yeah. So what are the key regulatory agency for biopharmaceuticals, especially Institution of Biosafety, IBSC board, Review of Genetic Manipulation, RCGM board, Genetic Engineering Appraisal Committee, and as well as Central Drugs and Standard Control Organization, CDSCO, and State Licensing Authority. These are the 5 authorities is responsible for reviewing and approval process of your drug approval in the respective markets in the Indian market. Yeah, the stages of the market, stages of the development. So initial stage 1, IBSC board, RCGM, and GS is the responsible. Product development, laboratory research till preclinical studies. And once preclinical studies is approved, clinical manufacturing, commercial manufacturing is approved by your CDSO board, CDL, SLA, ICMR, which is already I explained about this regulatory bodies is responsible for stage 2 development. And stage 3 development, life cycle management, handing off your post-approval changes and variation filing, CDSO, CDL, and state licensing authority is responsible for approval of your respect to life cycle management. And stage 1 product development, IBSC board, as we discussed, Institution of Biosafety Committee is registration required. RCGM approval also required and filing you're going to C1, C2 applications. RCGM application approval for import genetically modified organisms, GMO, LMO, living modified organisms, and followed by into Indian for R&D and activities form B1, B2 is responsible. And RCGM application or approval to conduct preclinical trials, C3, C4 application is required. And NOC for Form 29 license, test license to be issued for the manufacturing of the commercial products. And proof of concept studies, immunogenicity and safety vaccines from formulations is required part of your RCGM approvals. Conducting preclinical and toxicology trials, so required your respective authorities approvals. And RCGM approvals, application for approval of PCT and RCGM recommendation for you are going for clinical trials, Form C5 and C6 has to be approved. Genetic Engineering Approval Committee, as I mentioned, Commercial Activity and Environmental Protection Act, GESA approval is required. And what are the star mark is there? These are all mandatory for RDNA containing vaccines or biologics. Yeah, so current regulation framework for biologics and biosimilars in India. Stage 2, clinical manufacturing to commercialization, and NOC for Form 29 test licensing, how to file it, how to get it approval as a part of regulatory regulatory team and part of regulatory job responsibility to get the licenses. And Form 29 test license from State Licensing Authority, clinical lots manufacturing and CMC data generation, clinical trials phase 1, phase 2, phase 3 wherever applicable, and marketing authorization application CT21 application, and marketing approvals Form CT23 22 application, commercial manufacturing license application Form 27D, and joint inspection State Licensing Authority audit when they're coming for audit, CDSO and our State Licensing Authority jointly will inspect and give the GMP certification for the respective manufacturing site, receipt of manufacturing license Form 28D, and commercial manufacturing and distribution, and stage 3 life cycle management as explained previous slide. So these are the Indian regulatory authorities is involving in the respective filing pathways. Yeah, so now CTD structure, as I explained initially, there will be module 1 to module 5. When there is a called ACTD, there are in 10 countries in our world in Asia, there are 10 countries will follow in this format. So there is not much difference. So there is a 5 modules, here will be 4 parts, which is index is there in module 2, which will be not there in the ACD format. So your module 1, here part 1 remains same. Here module 3 will be part 2, module 4 will be part 3, and module 5 will be part 4. So this is the difference. Once you prepared CTD dossier, how you will convert into ACD dossier will be coming to know. Will be coming to know in our regular sessions. We will explain and we will teach you how to convert this CTD into ACD format. Yeah. And so the reference guidelines, M4Q guidelines, which is multidisciplinary guidelines. Q stand for quality guidelines for module 3 CMC, chemistry, manufacturing control, regulatory strategies, and data generation filing activities. So if you are attending for 45 hours course, you will come to know what is the M4Q guidelines and how to use this guideline as a guidance to prepare the module 3 CMC regulatory dossier preparation for the registration of your commercial products in the respective ICH regions and rest of the world. And refer to ICH M4S. M stand for multidisciplinary guidelines and 4 is the serial number, and your S stand for safety. Safety means your animal studies, preclinical studies. So which module 4 has to be refer preclinical trials, regulatory strategies, and data generation for filing activities. And refer ICH M4E guidance for module 5 and your clinical trial regulatory strategies and data generation for filing activities. So these are the main 3 guidelines to be referred and to be known regarding your filing pathways, quality, safety, efficacy. If you comply these 3 things, you can file your product in the anywhere in the world. If you not comply quality, you can't go safety studies. If your safety studies is not complies, even though you can't go for efficacy studies. So that this is the systematic way. Ensure quality, ensure safety, ensure efficacy. So this is the requirement for filing the product into respective markets. Then only your product can be filed, then only your product can be approved, then only can be able to market. Otherwise, you can't comply anywhere in the defective, anywhere in the not in compliance. Yeah. So preclinical stage, as we discussed, drug development, discovery, and your toxical studies will fall under preclinical. And clinical studies, phase 1, your pharmacokinetics, pharmacodynamics, phase 2, dose confirmation, phase 3, effective, and phase 4, marketing authorizations. So here there are regulatory bodies like there is a IND, investigational new trial, and IMPD, like in going for any EMA region, investigational medicinal work product dossier. In EU, this terminal will be used. The filing path will be remain same. CTA, so clinical trial application, amendments, anything. Clinical trial application, amendment will be takes place and followed by new drug submissions, biological drug application or new drug application or marketing authorization. Based on the region, based on the country, you can use these terms and words based on the filing pathways. Yeah. So this course can help the people like MSc Life Sciences, MTech, Biotech, or M Pharma, B Pharma, or B Tech, Biotech, or B Pharma, D Pharma. So the people from what are the background, life sciences background. So this course will teach how to R&D process can be do, and how to manufacturing, how to quality assurance QA jobs, and how to enter into QC jobs, and how to enter into regulatory clinical trials jobs. So these are the complete 5 modules, 5 regions, 5 places you can enter after completion of this course, and you can face R&D phase interview, manufacturing interviews, quality assurance interviews, QC interview, or regulatory interviews. So this is our contact details, and you can you may be already having the WhatsApp groups or maybe someone referred you. So you can contact us for the continuation of this course. So there will be a new batch starting from today is the demo session, and next week onwards we are going to start batch next coming Saturday onwards. Session time 2 hours to 2 hours and 3 hours on weekends. So in this weekend, Saturday and Sunday, we will conduct. So it may go around 3 months course, around 45 hours. So in this 45 hours, we will teach you entire thing, whatever the requirement for your regulatory affairs, or looking for R&D, looking for your quality assurance jobs, quality control jobs, or manufacturing jobs, as well as we will teach you complete internship completion followed by certification. So how internship topics for the enrolled candidates. So RCGM application filing for recombinant biological biosimilar products, one of the internship project, and RCGM application filing for preclinical non-clinical trial application, one of the internship project, and CDSO filing procedure for clinical trial application, one of the internship project. CDSO filing procedure for marketing authorization and application procedure, one of the project. IND filing pathway in US FDA, one of the project. You can choose it either one. And BLA, NDA, ANDA application pathway for in US FDA. You can choose your internship project. And MHRA filing pathways for UK registration of pharmaceutical drugs. You can choose one of the project. And EMA filing pathways for EU region. You can choose one of the project. And CTD modules preparation, module 1 to module 5. You can choose one of the project. CTD conversion into ACTD as we discussed previously, and ECTD uploadings. I will do it. This is the another one of the project you can take up. And RW markets, RW means rest of the world market, M1 preparation case study. So you already know how to file model 2 to model 5. Now model 1 is the difference for country to country. So that model 1 will be special case study will take from Saudi FDA. Saudi FDA is the very stringent, where it is required many data to file M1 approvals. So like this, there are 11 projects, and based on your interest, you can create any other new project also. There is no limit with this. When you come up with your in this anyone among this one you can choose, so that we can start internship. And end of the course trainee evaluation. So we'll have a one-to-one discussion, project review, respect to the among the topic whatever is chosen, rest of the project review, and followed by clarification will be issued, complete, and certification will be issued. The certification is from this institute, ISO certified institute. So we are going to mention internship project completion followed by certification issued. So upon your completion of your training, followed by project review and certification will be. So this is the related to internship topics for the enrolled candidates. Coming to what are the benefits of this course. So benefits for the enrolled candidates. Every session recording audio with video will be shared in the registered email through clouds. So access for watching at any time, and download option will be given. So you can download it and you can save in your systems for your future references. The every session. Today's session also record is ongoing. So post this session, I will give to your recordings. You can watch it. And every session applicable PPT will be shared in your same day through email for your reference. And every session relevant book or materials will be shared cumulatively, so that you can refer for internship project preparation, so that the respect to what are the topic you selected. So the respect to guideline or book will be given to you. You can start reading. So what are the going to give guidance Saturday, Sunday? You have to put efforts Monday to Friday and come back, and we'll discuss and will be guide you, so that your internship will be keep on running. So that within the 3 months, we can complete our internship project and respect to domain. And part of that, we'll be discussing complete CMC, and we're going to complete our sessions around 15 sessions in the 45 hours. And job opening and notification will be shared. So what are the WhatsApp groups are there? There will be, I am posting, keep on opening up the jobs. You can apply it, and CV review and suggestion will be provided. So we'll take your resume or CV, and we'll review it, and we'll give the suggestion to how to modify, and we'll give respect to CV suggestions, so that you can modify your CV. We can apply for the respective job. And technical mock interviews will be conducted. Let's imagine tomorrow is your interview. So we will take based on JD, known JD. I will ask the questions and you have to answer it. So when you're going to answer it, you can come to know that what are the gaps. When there is a gaps, so I will guide you. So what has to be prepared for that tomorrow interview, so that you will be very clear. So any gaps are there, we can be fill it for the tomorrow's interview. So we will be support for technical discussions and sessions at any time in the future as well. So it is not going to stop with 45 hours. So we can come up for any discussions, any guidance, any sessions. Will be support for any time in the future as well. There is no limit with us. There is no limit for any type of career growth and skill development discussions course. So this is the course will be support you for completion of internship as well as in future job search also. So only we are not giving any guarantee for job placements. This is in no one institute will give any 100% placements. If anyone is telling you in upfront, anyone is telling upfront, we are going to provide job placements means, let's imagine those are going to blame you, are going to your steal the money. That's all I can say that, because we don't know the after 3 months what is the situation. If I give commitment today, I will give the placement. If after 3 months, if I fail to provide placement, a single comment can erase 93 and 99 positive comments in the LinkedIn. So we are the open, and our posts also in the open the LinkedIn. You can go through the LinkedIn as well. So every post will be posted in the LinkedIn. If there are 99 positive comments are there, if one person put negative comment, so the 99 comments will be not be considered the people, only consider negative comment. So today for your benefit or my benefit, I can say that I will provide the placement. Let's imagine you paid the money and you already completed the course and already issued the certification as well. And after that, if I'm not going to provide the placement, so will you provide positive comment or negative comment? 100% you are going to put a negative comment. When you put a negative comment, so it will be damage for the institute. So for that reason, upfront and recorded session, I'm telling to you people. So this is a open and transparent communications we are giving to you, that we will provide assistance of the job, how to get it, how to enter, where is the opening, but there is no guarantee for placement. No one will give the placement guarantee. Yeah, that is the my side the today's session. Thank you all for the attending the session. If anything questions, let me know. We can spend another 10 to 15 minutes. Yeah, Ashutosh.
So I gave couple means whatever discussion happened, sir.
Okay.
It appeared like more of a theory, sir. Means what like practical knowledge we would be getting from this course, sir?
So see, this is completely related to regulatory affairs. So when regulatory affairs,
Okay.
practical knowledge, how to write the dossier, how to file into respective country. So this is completely overview of your regulatory affairs, not the process. The people willing to know process also can get the knowledge, can go for technical interview for the knowledge. So as per your internship expectation, you have to go to the laboratory and you have to see the how to run the HPLC system, how to run the purification systems or chromatography systems, how to formulation of filling systems. This is what you want to expectation. But when you want to go to the laboratory work, when you want to see the laboratory jobs, you can go to the that type of internship projects. You can go there, but this is 100% documentation activity. Even though you got the opportunity to work in regulatory affairs, you won't get opportunity to go in the inside the laboratory. Your job will be outside laboratory only, because your job will be limited with documentation activity, how to prepare the dossiers, how to gather the source documents, and how to align the filing pathways, filing documents. This is your job. So that is what you are going to get it, one type of direct exposure in the documentation activity for the regulatory job point of view.
Sir, I gave interviews for regulatory only couple of days ago. They
Okay.
were like asking means whether you have means like experience of softwares like Lawrence or Extido, whether you can label with softwares, other
Yeah, there is
regulatory softwares for documentation management. That's what they
exactly.
were more concerned. Yeah.
Yeah, so exactly for filing. Some of the countries, for example, FDA, so the sorry, US FDA, they asked to provide in the e-filing, electronic filing.
Hmm.
So there is a software is called Pharma-Ready. So I will give one of the presentation how it will be with the Pharma-Ready. The prepared dossier, how to going to upload into e-software. So when you know the CTD structure, then only can able to get idea on e-CTD for e-CTD submission. If you not know CTD format, very difficult to understand ECD submission. That CTD preparation also will get it, and your ECD uploading also you will get it in this course.
Okay, sir. One last question, sir.
Yeah, please.
If like we are supposed to deal with like medical device or cosmetics, would you
Yes.
also be teaching us how we would be handling the clients and all kind of things?
Yeah, so medical device is a part of our drug product, and cosmetics will be separate. Cosmetics will be not coming into our, because we are handling with our especially for the medicines, not with cosmetics. So cosmetics are beautifications and cleansing agents for the, that is separate division, cosmetics. But our is especially pharma and biotech. So chemical drugs, biological drugs only. So which is cosmetics not falling under this category.
Okay, sir.
Yeah, this is limited to chemical drugs and biological drugs, not to the cosmetics.
Okay,
Yeah,
sir.
yeah, yeah. Prajapati, do you have any questions? So Asutosh, shall we the session overview? Yes, Asutosh, shall we the session? Feedback. People can be text in the message as well. Yeah. Afipa, are you there, Afipa? Yes, Asutosh, you can talk. No one is talking, you can talk. Okay, thank you. So, any people have any questions, you tell me. Otherwise, I will ask one by one. Who are all there here? Around 15 people are there with me, along with me, 16 people. Prajapati, do you have any questions? Am I audible to you, Prajapati? Yes, someone real me. May I know your name, please?
This is Satish Durgam.
Okay. Are you there in the WhatsApp group?
I
And
joined. Yes, yes. Actually, I joined in the middle of the course. Accidentally, I joined. I
okay.
didn't know.
No issue. This session is recorded. So I will put, are you there in the WhatsApp group?
Yes, sir. I was. I'm there.
Yeah, I will post there. You can watch it the session. I will upload in the YouTube as well. And you can share that. I will share that video. You can watch it and starting from that. Yeah. How is your understanding? Thanks for joining.
You're welcome, sir. Yes, sir.
Yeah.
It's good actually.
Okay. Okay.
As
Any
I'm
questions
from,
do you have? Tell me. Tell me. Yeah.
so I'm from manufacturing side. So how this course will help me in what regard? So I just wanted to know.
How many years of experience do you have?
12 plus.
12 plus. Okay, this is for actually for initial starting level, entry level people. You test me separately, I will guide you. Okay, so there is a separate course for the experienced people, the people who are having experience in the manufacturing, research, and QC, how to enter in the regulatory affairs, and the people is who already in the industry for so long time and they want to become further enhance their skills. There is a
Yes.
career develop. Yeah, but there is a separate career development course. You text me in WhatsApp. I will tell you your name and your mail ID. You text me, and you mention how many years of experience and which domain, or even manufacturing, upstream, downstream, or formulation, whatever it may
Yeah,
be.
yes, sir. Sure, sure.
You text me clearly. I will suggest you which course will be fit for you.
Okay, sir. Thank you.
Yeah. Bhavana, do you have any questions? Afif, are you there? Bhavana, Karan.
Yes, sir. I would
Yeah,
like to
hi
know
Karan.
what is the costing of this course?
So this course for project complete internship will be 18,000. So every month there is a 3 months. Every month 6 months 6,000 has to pay. So by the time you are joining, you can based on the demonstration understanding. So we'll give the when you come up with your plan. So we'll give the what you can do that quotation will be given. This quotation can be showed in your college. You can get approvals if required. If not required approvals, you can strike forward. You can join it. And 6,000 has to pay for first month, second month 6,000, third month 6,000. So that means 18,000 overall cost. So will be whatever I mentioned in the recorded session in the previous slides, commitments, benefits that will be given all to the all for you. Yes,
Okay.
Darshan. Darshan, do you have any questions?
No, sir. I have one doubt for the internship program.
Okay, tell me what is your doubt?
Sir, what is the structure of internship and all other things?
That's what I mentioned previously, right? So what the structure means? In previous slide I shown. So this is what these are the internship topics. You can choose any one topic. So accordingly, the internship project has to be startup. So the course will be common for all. So if it's choosing even though IND filing, we will cover all the topics for our sessions. But your job for the preparation offline, you will be focusly on IND filing pathways only. And the person who chose MHRA, we will guide all the pathways in the regular sessions. But your role will be how to BLA application, how to IND application, how to end application. I will cover all the topics, but it's not possible to everyone to choose one topic, all the topics for your internship. So you can choose one of the topic. In that topic, you can become as a master. When you choose one of the topic as a become a master, when you mention your CV, you handle RCGM application in internship, so that when you go for interview, when you're asking about RCGM questions, you can crack that round of interview related to RCGM. This is your special topic. In every fresher interview, they may ask what is your strength, what is your which is the comparable topic. You can choose RCGM application filing, and you can tell that interview whatever you know, then easily can be selected in that interview. So these are the 11 topics, and not limited with the 11 topics. Anyone want indifferently, I can create for that internship project. These are the example I given 11 different topics for you. Yeah. Is it clear, Darshan?
Yes, sir.
Yeah. So you can come up, chosen, you can come back. Yeah. Mushraf Sheikh, do you have any questions?
No, sir. I don't have any question.
So with the session, Mushraf Sheikh, thanks for joining.
Yeah, it was great, sir. Very informative.
Okay. Okay. Yeah. So Prashant, do you have any questions?
No, sir.
Okay. With the session understanding?
Yes. It's good.
Okay. Preeti Mandati. Preeti, are you there?
Yes, sir. No, I don't have any questions.
Okay.
It
With the
was,
session understanding?
yeah, it was clear. I think it was more theoretical. Yeah.
Okay.
And
Fine.
I just have a quick question. Like, in the internship, like, you will be, are there any chances where you will be showing like how we have to use the tools and like publishing tools or whatever the
Filing pathways definitely will show. For example, I will show the based on your interest, I will show it how to show it. So if you go to the for example, you want to file any one of the product into RCGM. So there is a portal, Sugam. Or you can take it IBKP portal. Yeah, my screen is visible. The Google Chrome is visible.
yes.
Yeah, visible.
Yeah, so this is the live. In generally, the organization will having login IDs. When you log in here, you can enter into login credentials. It will having only organizations. It will not given to everyone. Even though our institute also don't have it, but I know the process how to apply here. For example, there is a IBSC. There is a institutional biosafety committee, or when you want to file any IBSC application, or you want to file RCGM application, click here. So there is a clear guidelines, clear requirements, checklist, everything is mentioned. So in our regular sessions, respect to session and respect to day, so which we are talking about regarding, for example, you are talking about pre-clinical approval application, how to enter here, how to apply, and how to prepare the application, I will tell you. So even though you got job in the regulatory affairs, at the same time, you are going to do also same activity, but in that case, when you join in your organization, you will have a login credentials. You can go there and you know the offline activities, all those things, what has to be prepared. Once you log in, upload the respective documents and submit it. That's all. So you will get maximum knowledge. 99% you will get it. 90% knowledge you can get it from offline from our institute. 10% the moment you are entering into industry, you can handle it. So this is what you are going to get it in our institute. Yeah, is it clear?
Yeah, it's clear. Thank you.
Yeah. So like this, you can get the knowledge. So like this, not only limited with your IBKP portal. So there is a called one more portal is a Sugam portal. So which will be guide us how to apply clinical trials. So if you see here, apply online for new drugs, subsequent new drugs, investigational new drugs. This will help us. And there is a apply online for fixed dose combinations. There is a RNA derived drugs. So when you log in here, Sugam portal, when product is belongs to recombinant products, you have to enter in this pathway. And products belongs to blood products, plasma products. So today, these are also one of the product portfolio where you having multiple blood products. How to apply here? We can get it easily. And dual use, no objection certificates. How to get it? Apply online licenses and get the approvals. Where you are getting approvals from India, you can get it throughout the world submissions. And related to state FDA manufacturing site approvals, inspections. So that will be also state-wise will be having a FDA that will come for visit through CDSO. And apply online for vaccines, biologicals, how we will get it approvals. And related to registration on open for clinical trials, how you put application. In Sugam, online licenses, this and all will get it. So this is what we are going to teach you. We don't have login IDs, but I will teach you how to prepare the application, how to place it, respect to domain. So respect to base, if you select the project related to RCGM, straightforward, we'll go to the IBKP portal, we'll go to the RCGM, and I will teach you how to get it here. Yeah, this is what we'll do.
Yeah, sure. Thank you.
Yeah. Any other questions, Priti, from you?
And in the internship product, like you will be going through the post-marketing authorizations and how will be the recalls, complaints.
Yeah. So there is a products like insulins, and we are going to products like maps, products like vaccines. These are the case studies we are going to conduct. And this registration process, even though you select your internship project is related to RCGM, not limit with this. Our session will be continue all three products start to end. Start means how to start R&D, how to optimize at R&D, how to technology transfer, how to register into commercial scale up, and followed by commercial submission. That will cover. So you have to look for your specific domain RCGM filing activity and as well as other product registration, and not only limit with your product filing in the limitations respect to variation filing, respect to recalls, respect to annual product quality review, all will be covered. There is a QMS session. In that session, we'll cover product recalls as well as annual product quality review. Yeah, that will be covered there.
Yeah, sure. Thank you very much.
Yeah. Anything else?
Yeah, that would be all. Thank you.
Okay. Yashaswini? Yashaswini, are you there? Okay. May I know this Zoom username? Okay. So based on your interest, you can ask any further question also. I'm going to stop the recording, and after the recording also we can discuss. There is a question. There is a Yashaswini. No answer. No question. Okay. Thank you, Yashaswini. Yeah.